Welcome back, everyone, to the first session of the conference. We have four talks in this session and a poster session in between. So, our first talk is by Samuel Awini. He's going to talk about the dynamics of forest elephant crop breeding, talking about and continuing to talk about camera traps, DNA, and dung metrics. So, Samuel, the floor is yours.
All right. Thank you very much for this opportunity. My name is Samuel Awini, as introduced, and I'm going to talk to you about the dynamics of forest elephant crop breeding and insights from camera traps and DNA dung metrics methods. So this study was undertaken in Gabon, in Central Africa. And as we all know, human-wildlife conflict is on the rise, predominantly overtaken by elephants due to their larger body size and the distractions they are capable of causing within the shortest possible time. And Gabon is a home to probably half of forest elephants population in the entire world or in the entire Africa where elephants, the forest elephants occur. So this study was really important to have undertaken in that place. And poaching or habitat loss is a cause and also a threat for this kind of activity. The forest elephant was recently classified as critically endangered, leading to or making it more important for such a study to occur. The aim or the objective of this study was to define a population profile and behavior of forest elephants involved in crop breeding. And my objectives was to define a population demography for all elephants or crop elephants that are involved in breeding, especially get to know their sex, their age, the group size of individuals involved in crop breeding. I also wanted to quantify the frequency and timing of elephants, at which they visit the farms to raid crops. And I also wanted to compare a number of elephants and then the timing of elephants or the times at which elephants visit farms and for raiding, and also the times and numbers at which they stay in the forest, which is their natural habitat. As mentioned earlier on, my study area was in the Gabon, the southwest, the part of Gabon in Central Africa, in the national park, a protected area, which is the Mukalaba Dudu National Park. It covers an area of 4,500 kilometers square, a rainforest, but having significant patches of savanna habitat. The area is bordered by several villages, but human population in those villages is minimal. And most of all these villagers or people are into crop farming for sustenance, which is highly impacted by crop breeding. So it was important we get to know what is happening and how we can resolve this issue. This study area has a higher elephant density, just like any part of Gabon, and there are lions, buffaloes, chimpanzees, and several other animals which all involve in or are all involved in crop breeding. But elephants causes most destruction, like I mentioned earlier on. So my study was in two sites. The first one was in a crop farm, which was a crop farm. And the crop farm, I divided it, or it was in two plots. The first plot was demarcated to be a new farm, which had, there was still an active cultivation, meaning there were farm crops, and the farm was protected somehow to prevent elephants from entering and causing destruction. And the other hand, or the other side, was a farm that was abandoned. It was farmed several years ago and were no more under use or protection. So there were no um, elephant protection mechanism on this farm. And most of these protection were metal wires hanged around the farm, serving as a fence with noise makers like empty cans and metal sheets hanged on them to scare or deter elephants from entering the farm. As you can see from the map, I have the old farm and then the new farm with a red line serving as a boundary, and also which is also a metal wire fencing the old farm and the new farm. So the new farm, or both farms, have both the old and the new still have some farm crops, but much attention, or most of the attention, is given to the new farm. And you can see from here we have cassava, sugarcane, and several other farm products or crops that are being cultivated in these farms. An overview of my study area, you could see Gabon, and then Calabar Dudu National Park, and then a forest habitat, and then the farm area or the farm site where the studies occurred. So the study methodology, I collected data using camera trapping, DNA sampling, and then I also measured dung circumferences of elephants. And the aim was to identify sex, determine age. So I wanted to identify individuals from the cameras and also from the DNA samples, and also determine sex from cameras and also from the DNA as well, and also estimate age of the elephants involved in conflict from the camera traps and from the dung measurements, and also determine the time of visit and number of individuals from the camera traps as well as the dung measurements. So in all, I used 30 camera traps in the forest, covering 15 transit lines, covering 10 transit lines, and then 15 cameras covering 4 transit lines in the farm. All cameras were set to take only pictures. And then for DNA, I surveyed both the forest and the farm to collect fresh DNA from fresh elephant dungs, which I guess to be at least not more than 24 hours old. And then I went ahead to measure, after taking the DNA, I measured the dung circumference of the largest dung body of each dung pile to estimate age of the elephant as well. And for data processing, I then used Camlot to sort and tag individuals to get time and date and events when the crop reading occurred. And then we went ahead and extracted our DNA from these dung samples to identify sex and individuals of the elephants involved in crop reading and those in the forest as well. I then used these papers and this book to estimate elephant age based on the mean of the dung measurements I took as well. For the camera trap data, I took in the forest, the total of 28 camera traps efficiently worked. You know, I used 30, but 28 were able to produce results. And in the for the forest, and then in all, I had a total of 182 and 676 total photos of elephants, of which 68 were independent elephant photos and from the forest, and then 248 were independent photos of elephants from the farm. Based on sex, based on individual elephants, the forest I had 94 individuals. I was able to identify 94 unique individuals from the forest, from the cameras, and also from the farm, 392. And for sex, we had 56 males from the farm, and then females 39, and 158 were unknown. I wasn't able to determine their sex based on the camera from the farm. I didn't do this for the forest because my interest was on crop breeding, which is in the farm only. And similar results was also obtained for the age groups as well. For DNA and dung metrics or measurements, I collected a total of 213 DNA samples from both forest and the farm, 96 from the forest and 117 from the farm. And a total of 184 dung measurements were made from these out of the 213, because in some dung piles, I couldn't find a complete or a more cylindrical or intact body to measure. For genotyping or for DNA extraction and genotyping, a total of 187 samples were able to or passed through the genotype, and then we obtained a complete genotype for 169 samples. And we identified 92 from the forest, 92 in total, and 57 from the forest, 48 from the farm. For the forest had a recapture rate of 0.456 and the farm 0.7, which was really higher. And in terms of sex, the farm had a total of 29 females and the forest had a total of 19 as well. For results and discussion, so in the farm, the camera traps were placed around the boundaries of the farm along a transect running through the farm. So I then went ahead to check how these cameras were spaced or distributed by observing or recording the movement direction of the elephants. Either they were entering the farm or exiting the farm. And I saw no significant difference between the number of elephants entering the farm and that of those exiting the farm, which means that we had a uniform coverage and also uniform or a balanced movement of elephants to and from the farm. For the profile of the demography of the crop breeding elephants, we saw the majority of these rates or majority of the crop breeding were caused by males from the camera traps data, and it was adult males followed by sub-adult juveniles and then sub-adults. But majority of the sex of these elephants were unknown from the camera traps because it was difficult to determine the sex from an image. Mostly the reading was caused at night, so you couldn't tell that this elephant is a male or female, but you could tell if it's just huge, then you say it's an adult, but very little can be said about sex. And for the group size of individuals rating, we saw that most of them done by individuals alone or lonely individuals. But sometimes we saw up to a group of 4 involved in raiding as well. And there was a strong or significant association between sex and age groups in raiding as well. But for the DNA profile, it was quite different. We didn't see any significant difference between the number of males and females involved in breeding, but there were still adults involved in breeding or causing most of the raid. And there were no significant difference between our association between sex and age of elephants involved in raiding. So the DNA data somehow contradicts the camera trap result or data. Based on time and visit patterns, out of the 46 steady days or period, elephants visited the farms or raided the farm 45 days out of the 46. And 7 out of 7 of the weeks we did, even though there were variations, but it was constant. They visited the farm each and every week as that. I then went ahead to look at individuals that were frequent or most frequent and classify them as habitual raiders. And I saw 9 individuals that has visited the farm at least 3 times. At least 3 times to raid the farm. And out of these, for these 9 unique elephants, 18.7%, they formed 18.75% of all the 48 unique elephants observed, and they were responsible for 45% of the total raid that occurs in the area. So only 9 elephants causing 45. That was huge. It means they were always around. A number of elephants visiting the farm versus the number of elephants visiting the forest. We saw a higher number of elephants in the farms compared to their natural habitat, which is the forest. And that could be from the camera trap data, and that could be because of feed. We have ease access of food in the forest, in the farm compared to the forest as well. But we did not see any difference for the DNA data as well. And for timing of visit in the farm, we saw a clear difference in elephants visiting the times they visit the farm and the times at which they visit the forest. We observed that elephants visit the farms at early morning between 6 a.m. and 7 a.m. and then later in the night from 18 hours to 23 or 22 sometimes. But for the farm, for the forest, the elephants were mostly active only in the daytime, which was a very smart move in my opinion. They were actually the ones controlling the conflict here or managing the conflict here, since they knew when to avoid going to the farm, because they know that during daytime probably there are a lot of people in the farm, so it wouldn't be convenient to raid. So yeah. So for the strengths and limitations of this study, the camera traps were somehow efficient in telling us the time, the numbers of elephants involved and all that, but it was difficult to determine the sex and age of elephants involved or even found in the picture. From this picture above, you could see it was taken at night. There are elephants in there where I've circled red, but you can hardly see them or even get to know their sex or age from this picture, which was a limitation for using the camera trap. And for the DNA, I was not so sure if DNA, the sampling had covered or were able to efficiently collect all elephants that had visited the farm or recorded all elephants that had visited the farm, because sometimes elephants could visit, raid, and go elsewhere and defecate. They don't have to necessarily leave their dung where they have raided unless they spent a lot of time there. So if I go to the farm to collect just samples from the farm, I may not have collected everything or all individuals that actually caused the raid. And the season at which I did my study was also in the rainy season, which also affect or impact the distribution or movement of elephants as well. But the major strength of this study was the application of the various methods, the camera traps, the DNA, and then the dung metrics, each of them complementing the other to strengthen the results or the data I obtained. So in all, I would say that the current mitigation strategies used in the farms, which is using the metal wires, and then hanging empty cans on it, is somehow effective because we only saw elephants raiding the old farm crop, or I only found dung or DNA samples from the old farm area compared to the new area, because probably because they get enough food in the old area so they don't have to go to the new farm, or maybe the wires are just effective and actually prevents them from going to the farms. And recently, the government also approved killing of elephants that are habitual raiders or causing trouble or threatening life or distraction to people. And I'm not sure if from these results, killing elephants would actually solve the problem, because we saw that juveniles and males and females, there's no clear pattern. Every elephant is actually involved, so you cannot actually select to kill to solve this problem here. And ethical considerations as well. So in the future, probably it would be nice to track some of these elephants to actually know their movement patterns, since the evidence left behind after reading, which is the dung or pictures, may not actually or may not always be available. Tracking these elephants would actually prove very useful in the future, since we'll actually get to know where they go, what time they are there, and etc. So in conclusion, human-elephant conflict in the area involves several complex factors rather than just male elephants, or maybe just females, or maybe adults or juveniles. It involves a lot more than that. So a holistic understanding is required in order to mitigate conflict in the area. Thank you very much for your time, and I'm around for any questions you may have.
Thank you, Simon. A very fascinating talk. I really liked it. We are running a little late, so maybe we have time for one quick question from the YouTube chat. So Valentina Bar asks, how did you store the dung samples for DNA analysis?
So I used light and airtight DNA sampling kit or container. So I collected samples in the field using the swabs, buccal swabs, and then store them in a buffer, lysis buffer, and I mostly keep them for about 2 weeks maximum, and then immediately transfer them to the lab for extraction and storage under less than 20 degrees for a long time.
Thank you so much. I think if there are more questions, you will be around on Discord to answer, I guess.